Hello, I'm Milton McAllister. I'm a veterinarian and a scientist with the University of Adelaide, and I've been asked by Cambridge University Press to discuss this topic about vaccines that we have in veterinary medicine that control diseases of animals that are caused by malaria-like organisms. So what do we mean by malaria-like organisms? Well, these organisms are protozoa, which are larger than bacteria, which are small and simple in comparison. Protozoa, although still being microscopic, contain internal details such as a nucleus and mitochondria. And all of the examples that we're going to discuss now include the apical complex for invasion into animal cells or human cells that they parasitize. And they replicate inside of those cells and then bust out. So every organism we're going to talk about has this basic structure and that basic life plan. So let's compare vaccines. There are no vaccines for malaria or any other protozoal disease of humans, but there's a candidate vaccine that is close to being licensed. It requires 3 inoculations, and it induces immunity in about half of vaccinated people that endures for about 6 months. In contrast, all of these diseases of cattle and sheep have vaccines that induce solid immunity in the great majority of animals and endures for much longer than a year, often for the life of the animal, from a single inoculation. So what accounts for the great difference? These animal vaccines contain modified forms, weakened forms, or controlled forms of living organisms. The malaria vaccine only contains little bits and pieces of malaria organisms, DNA and protein. There's a great desire for any human vaccine to be very safe. The problem is that although these vaccines are very safe, they haven't been very effective. And in the meantime, hundreds of millions of people have been developing malaria every year, and someone has been dying of malaria every minute. So it seems that there is a good case to go ahead and use the classical techniques of using live organisms in a malaria vaccine, and new emphasis could be placed upon developing new, modern, novel ways of making those live vaccines extremely safe.